After the introduction of gene therapy, next is the types of gene therapy. So there are two types of gene therapy, somatic and germline. So as the name is showing, like in somatic gene therapy, the therapeutic genes they are inserted into the somatic cells, and in germline gene therapy, therapeutic genes they are transferred into the germ cells. So, what kind of cells we target in somatic gene therapy? They are like bone marrow cells, blood cells, skin cells. And in germline therapy, we target sperms and eggs. So, being into somatic cells, these genes they are not inherited. So, they are not inheritable. And being into the germ cells, the introduced genes they are also inherited in the daughter progeny, so it shows heritable pattern. And at present, like all the research that is going on in this somatic gene therapy, and for safety, technical and ethical issues, this germline gene therapy it's not being currently targeted. So what we will be focusing is on somatic cell gene therapy, in which the genes they are inserted into the somatic cells. So this somatic cell therapy, or it is, there are like two type of somatic cell gene therapy, or we can say there are two type of approaches or two methods of somatic cell gene therapy, ex vivo and in vivo. In ex vivo gene therapy, the genes they are transferred into the cells but outside the patient's body, and in vivo gene therapy, the genes they are transferred directly inside the body, obviously in combination with some liposomes or plasmids or some vector. So this is a generalized or a schematic representation of both kind of approaches. Like here the genes they are taken, here the cells they are taken from the patient's body. In ex vivo approach, the cells they are taken out from the body, and the therapeutic gene that is inserted into the somatic cells, the cells which take up the gene, therapeutic gene, they are selected and amplified, and then they are put back into the patient's body. So this is ex vivo approach. In vivo approach, what we do, we just take the gene directly, put it in some vector, and that vector is introduced into the patient's body directly, and that gene, that carrier will carry the gene into the target cell or tissues. So next is we will study in a bit detail about these both these types. Ex vivo gene therapy, its example is best shown by ADS kit, that's adenosine deaminase severe combined immunodeficiency syndrome, the same one that was when the first gene therapy was attempted on a 4-year-old girl, Shanti De Silva, who was suffering from ADS kit. So that was ex vivo gene therapy. That means her somatic cells they were taken out from the body. So in this mode of gene therapy, the genes that are transferred into the cells that are cultured outside the body. Then the cell is transformed, or we can say it is transfected. The transfected cells they are selected, multiplied, amplified in vitro, and then they are put back into the patient's body. The cells that we target in somatic gene therapy are generally the bone marrow cells and skin cells. They are the general cells that we take in for somatic cell gene therapy. And after the establishment of human embryonic stem cell lines in 98 to 2002, these can also be used for this type of therapy, ex vivo therapy. So the main advantages being autologous, being from the patient's own body, they avoid any kind of immune rejection, and gene transfer efficiency is high when we use retroviral vector for transfer of the gene, and we can enrich the cells that have taken up the gene. We can enrich the transfected cell using selectable marker, and the transfection efficiency that can be evaluated before putting it back into the patient's body. So these are the advantages of using ex vivo gene therapy. And what are the main disadvantages? Like it being because we are using here RV vectors, retroviral vector, so they target only the proliferating cells, dividing cells, and tissue culture method is also required. That means we should be able to culture in vitro the cells, the target cells, we should be able to culture them in vitro, and transplantation technology should be there. that means how we are going to put back the cells in the patient's body, that technique should be there. And this is again a schematic representation of the ex vivo gene therapy approach. So we can have a look briefly, like the cells the cells which are defective in gene, they are harvested from the patient's body, they are cultured in vitro, and then they are co-infected with the retroviral vector that are containing the therapeutic gene. So here this therapeutic gene, it is packaged inside the retroviral vector, and then this retroviral vector is co-infected with the target cells that are already that we are already culturing, and then infection is infection like then the cells gets infected, that means they get transfected. Then the transfected cells, they are selected using selectable marker, then they are amplified further in vitro, and then these amplified these cells which are carrying the therapeutic gene, that put back into the patient's body. So ex vivo gene therapy. Next is in vivo gene therapy. In this mode of gene therapy, the therapeutic gene, that is packaged into a vector simply, and then it is directly transferred into the target tissue or the cell in the patient using a syringe. And if we are given a chance, if both type of therapy are possible, we prefer for ex vivo gene therapy. Okay, so but it is more common and more certain, we are more sure of results in this in vivo gene therapy, and it is done, generally it is done when we are not able to culture the cells, patient cell outside the body, or when there is reimplantation technology is not efficient. For example, brain cells, it is very hard to culture them under in vitro conditions. So for to target these kind of tedious cells, so we go for in vivo gene therapy approach. And in this gene needs to be delivered only in the target cell, and the retrovirus delivers the cell only to the dividing cell. So this could be one of the limitation of in vivo gene therapy approach, the same one that we get to see in the ex vivo gene, because these are the kind of viruses that infects only the dividing cell, but these can be efficiently utilized for targeting of cancer cells. And in other cases, we can for RME, that is the receptor mediated endocytosis, or we can insert tissue specific promoter upstream to our therapeutic gene, so that their expression occurs only and only into the affected tissue or the cells. Disadvantages are, as compared to ex vivo gene therapy, specificity is less, and efficiency of stable gene integration, it is also less as compared to ex vivo gene therapy approach, and there is always a tendency for host immune rejection. So this is again a schematic diagrammatic representation of the in vivo gene therapy. So in this is a therapeutic gene that is inserted into the either into the virus, retroviral virus, or the liposome. These are the lipid bilayers, synthetic lipid bilayers, into which gene is packaged inside, or the plasmid. So either of these three is taken as a carrier or as a vehicle or as a vector, and then these genetically engineered DNA, that is inserted into the patient's body by direct tissue injection. So the inserted DNA that gets into the specific cell and produces the needed protein that is encoded by the inserted therapeutic gene. So these are the brief steps. So it is packaged into these, and these are put into the patient's body, and the patient the disease, it is recovered by the expression of the relevant protein for which therapeutic gene is coding. So next is the steps of in vivo gene okay, so these were the steps. Next is the vectors for gene delivery. So now we know what is gene therapy, what are the main approaches of gene therapy. Like there are 4 approaches: gene replacement, gene silencing, gene augmentation, and suicidal gene. Okay, so after that we studied about the two type of gene therapy, somatic and germline gene therapy. So germline gene therapy is prohibited right now, being technical, safety and ethical issues. Somatic gene therapy is the gene therapy that currently we are using all over. So this somatic gene therapy is further divided into two type of somatic gene therapy, that is ex vivo, in vivo. When it is occurring outside the patient's body under in vitro condition, that is known as ex vivo gene therapy. When it is occurring inside the patient's body, inside the patient's cell, that comes under in vivo gene therapy. So after these types of somatic gene therapy, next is the vectors which we are using for delivery of the therapeutic gene into the cells, maybe inside the body or outside the body, ex vivo or in vivo. But what are the main vectors or the carriers that we are using for the transfer of therapeutic gene? So there are two kind of vectors. Either it could be viral vector or non-viral vector. So a viral vector includes adenovirus, retroviruses, adeno-associated viruses, lentiviruses, vaccinia, and herpes simplex viruses. Out of these first three, adeno, retro, and adeno-associated viruses, these are the more frequently used viral vectors. And non-viral vector includes the lipid complexes, liposomes, protein, peptide or polymers, and physical transfection method like electroporation and ballistic gun particle bombardment method. So these are the non-viral vector which we'll be discussing in the next clip.